Hi, a very good evening to Dr. Siti Farah. My name is Ong Sing and my metric number is 181441. So based on the first question, state briefly how you are going to develop a blockbuster drug if you are the director of the pharmaceutical company. First of all, to come up with a new drug, you have to find a drug that is actually they may treat a disease that affect a big populations or first world countries such as the cancer, diabetes, obesity and others. And it also may show, the drug may show no or less side effect or adverse effect, and it may susceptibility to most of the population. So to develop a drug, it should be started from a drug discovery to know the mechanism of the actions of the disease, and then to the design a product to reverse or to stop the effect of the disease. So the drug can be in the many forms, endogenous, either in endogenous compounds, synthetic drug, or even from the nature. And first of all, the lead compound should be identified first through the high throughput screenings or computers aided drug designs, or through the serendipity to identify a lead compound that actually there are possible beneficial the effects of a large numbers of the disease. And after that, by knowing the promising drug that to develop, after that will be conduct experiment in the different assay such as in vitro, in vivo and ex vivo, and then to determine the toxicity and the efficacies of the in the animal models, and to also to determine the pharmacodynamic and pharmacokinetic of the drug. After the preclinical trial in the animals, it should be further into the phase 1 clinical trial and continue with the phase 2 and phase 3 in the human to test whether it's safe and effective in the human or not. So for the next question is to explain your strategy to make sure that the drug can become the most potent drug. So how the drug to be most potent? First of all, you have to knowing the potential benefits and the mechanism of the actions of the drug, and then the best dosage and the best way to administer of this drug, either through oral injections or inhalations, and also know how it interact with the other drug and the treatment, and also can be know their toxic dose, effective dose and the later dose through the dose response relationship graph. And actually structure activity relationship also play an important role here. Is the method to determine or to identify drug target interactions that may enhance the activity of the drug. So the drug is designed to specifically target and to react with the specific biomolecular such as the nucleic acid, proteins, enzyme or receptor and so on. And for example of the drug target interaction such as the hydrogen bonding and the hydrophobic interaction. For hydrogen bonding, the hydrogen bond donor, they may have a highly polar hydrogen atom that is bonded to the hydrogen bond acceptor, which have a strongly electronegative atom, primarily in the oxygens and the nitrogen. And after beside that, actually if too large or absent of the group in the drug, that may not fit in the active site, and the compound is inactive to show the effective to the disease. Next, it should be also know the physiochemical properties of the drug molecule as well, such as the pH, acid base, and related acid strength, lipophilicity, and other, to determine or to control the absorptions, the eliminations, or the permeability of the drug inside of a body. Okay, I think that's all from me.